polymerase chain reaction or PCR. It is a test to detect genetic material from a specific organism such as virus.
Restriction fragment length polymorphism, or RFLP, is a hypopolymorphism that results from variation in the DNA sequence recognized by restriction enzymes. These are bacteria enzymes used by scientists to cut DNA molecule at known location. DNA sequencing is a laboratory technique used to determine the exact sequence of bases, A, C, G, and T, in a DNA molecule. The DNA-based sequence carries the information a cell needs to assemble protein and RNA. Molecular DNA sequence information is important to scientists investigating the function of genes. Cases of prenatal or perinatal diagnosis of a birth defect or an enzyme deficiency is one situation where DNA diagnosis could be of immediate beneficial use. What must be emphasized is that the information to be gathered by the new technology should be private and used by choice by an individual whose DNA has been read. In a nutshell, a person's DNA sequence, to my mind, should be protected by law from distribution to other parties without the individual specific permission. This goes the same with the physician who have obtained such information, who should also treat as they do any privileged material.
In my opinion, it is about information that should not be open to public. It should be private as possible. The patient information must be confidential and can only open the public if it is authorized by the patient.
The introduction of foreign genes into the organism doesn't seem to be dangerous. The resulting organisms don't appear to be better able to reproduce than any others. Attempts to remedy human disease by somatic gene therapy also don't appear to pose ethical dilemmas, but the introduction of genes into the human germline does. The diagnostic procedures that genetic engineering allows are very powerful. Before anything else, it will be relatively easy to correlate the presence of various DNA polymorphism with disease and other heredity characteristics.
Reduction of foreign genes is that we should not be afraid of it. It is said that does not affect human life or any organisms. It is used to remedy human disease.
In genetics, expressivity is the degree to which a phenotype is expressed by individuals having a particular genotype. Alternately, it may refer to the expression of a particular gene by individuals having a certain phenotype. Expressivity is related to the intensity of a given phenotype. It differs from penetrance, which refers to the proportion of individuals with a particular genotype that actually express the genotype. Variable expressivity refers to the degree in which a genotype is phenotypically expressed. For example, disease can have the same genotype, but one may express more severe symptoms, while another carrier may appear normal. This variation in expression can be affected by modifier genes, epigenetic factors, or the environment. Modifier genes can alter the expression of other genes in either an additive or multiplicative way, meaning the phenotype that is observed can be a result of two different alleles being summed or multiplied. However, a reduction in expression may also occur in which the primary locus where the phenotype is expressed is affected. Epigenetic factors such as cross-regulatory elements can also cause variability in expression by inducing variation in transcript abundance.
Genetic engineering. Gene therapy involves altering the genes inside your body cells in an effort to treat or stop the disease.
It involves the removal of a desirable gene from a cell, adding it to the gene structure of a bacterial cell and replacing the recombinant DNA into the bacteria cell. The bacteria then have the capability to produce the protein produced by the original animal or plant cell.
In 1994, Calgin's delayed ripening tomato became the first genetically modified food crop to be produced and consumed in an industrialized country. Since the recorded commercialization of GM crops in 1996 to 2018, several countries have contributed to 113-fold increase in the global area of transgenic crops. The area of planted crops shot from 1.7 million hectares in 1996 to 119.7 million hectares in 2018, with an increasing proportion grown by developing countries. In 2018, there were 26 biotech countries, 18 of which growing 50,000 hectares or more, 21 developing countries, and 5 industrial countries. They were in order of hectareage: USA, Brazil, Argentina, Canada, India, Paraguay, China, Pakistan, South Africa, Uruguay, Bolivia, Myanmar, Sudan, Mexico, Australia, Philippines, Spain, Colombia, Vietnam, Honduras, Chile, Portugal, Bangladesh, Costa Rica, Indonesia, and Eswatini, ISA 2018.
Genetic engineering is a process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring which desired traits. Genetic engineering involves the direct manipulation of one or more genes. Most often, a gene from another species is added to organisms' genomes to give it a derived phenotype.
Genetic engineering is a term that was first introduced into our language in the 1970s to describe the emerging field of recombinant DNA technology and some of the things that were going on. As most of the people who read textbook and things I know, recombinant DNA technology started with pretty simple things: cloning very small pieces of DNA and growing them in bacteria, and has evolved to an enormous field where whole genomes can be cloned and move from cell to cell. To still using variations of techniques that all would come under genetic engineering, broadly defined, means that you are taking pieces of DNA and combining them with other pieces of DNA. This doesn't really happen in nature, but is something that you engineer in your own laboratory and test tubes, and then taking what you have engineered, propagating that any number of different organisms that range from bacterial cells to yeast cells to plant and animals. So while there isn't a precise definition of genetics engineering, I think it more defines an entire field of recombinant DNA technology, genomics, and genetics in the 2000s.
This schematic illustration shows how a human therapeutic gene is inserted into a deactivated mouse retrovirus. The retrovirus then attaches to an empties its genetic material into a patient's cell in the laboratory. The therapeutic human gene is integrated into the patient's DNA and replaces the defective gene in the treatment for ADA.
Genetic disorder can be the result of genetic abnormalities, such as gene mutation or additional chromosomes. The effects of abnormalities is an individual DNA were once entirely unpredictable. However, modern medicine has produced methods of identifying the potential health outcomes of genetic disorder, as evidenced by medical research from educated, advanced degree nurse practitioner and practicing physician, collecting the following evidence-based statistical observation. These professionals have identified some of the current best practice for detecting, treating, and potentially preventing some genetic disorders. Disease people cured by fixing their genes. Putting into plants, domestic laboratory, animals is one thing. What about using genetic engineering to modify humans, in particular to cure humans' disease? There are two quite different procedures that are being debated under this heading, and they need to be discussed separately. The first procedure is to insert foreign genetic materials into the line, into sperm or eggs, where they could be passed into a succeeding generation. The second procedure is to use genetic engineering to insert foreign genes into somatic cells, into any cells except sperm or eggs and their progenitors in an effort to correct some genetic defect.
Some question may arise like, does the process work? Will it yield the better of life or the treated individual? When the patients or guardians given informed consent, treating human genetics by purposely changing the genes that are passed on to succeeding generations seems potentially dangerous and ethically unsound. It is dangerous precisely because of the problems brought up at the very beginning. There is a great possibility of some great interaction between one introduced gene and the remainder of the genome. The consequences of this ignorance fall into succeeding generations. On innocent children, our fetuses. On the other hand, if the genetic engineers and the physician make a mistake in somatic therapy, the person who suffers and the patient, who should have appraised of dangers before the therapy was begun.